Hello, I am sorry that I'm not here with you today, but you are going to have to learn through an audio recording of what the lesson would be today if I was in class. And Ms. Darnell is there to help you if you have any questions along the way. I hope that you all survived the QA yesterday. I'm assuming I'm hearing some groans right now. That's okay. I will get those graded and back to you as soon as I can. We're going to do an extension of what we did on Monday with the gene machine. So I wanted you to think back to that lesson where we played out the skit with the gene machine. What does it do? You're right, it does do a lot of those things. It's not enough to know the patient's DNA sequences from a genetic test. A genetic test must compare the patient's DNA sequence to known sequences. These known sequences are what we call reference genes. You might want to take a few notes today, just over DNA databases. We need to know the patient's DNA sequence, but we also need to have something to compare it to. We call this a reference sequence. Databases allow scientists to store, manage, and retrieve information in an organized way. Think kind of about like how if you use iTunes, how it works. There's a lot of information out there, though I don't use iTunes, so I don't really know how it works. But there's a lot of information out there, and there's a way to store information, manage it, and then if you want it later, to retrieve it back. And that's what scientists are doing. They create these databases, these gene databases, store genetic information, to manage it, organize it, and then ultimately retrieve it for later use. There are lots of DNA databases out there. One of the ones we're going to be exploring today is actually called, in my page here, NCBI, which is a National Center for Biotechnology Information. It's actually part of the National Library of Medicine. Which is actually part of the National Institute of Health. So it's a public domain. It's information that's out there. It's available to the public. NCBI actually houses information in over 30 databases. So it's not really just one database. It's a collection of databases. It's all related to genetics and molecular biology. All of these databases can be searched using one complete search engine. It's called INTEREST. Items stored in different databases are cross-referenced and interlinked, making it easier to find all the database records that are related to any one subject. We're primarily going to be using the nucleotide database today. So I'm just going to pull up an image of this. This is NCBI. You're going to be visiting this here in a little bit. You'll notice that if you go up here, all databases, there are over 30 databases in here that you can search. If you type in this bar up here, this is going to search all of the databases together, not just one of those databases for whatever information you're looking for. There's all kinds of resources here. Some of these you'll get a chance to just kind of poke around at and look and see what they are. We're going to be using something called BLAST later on, and that's something we're going to use on Friday, but we're also going to be using when we get into our evolution unit. So you'll kind of have a chance to just kind of poke around here and see what there is on this website, and then you'll get a chance to look for something specifically I want you to look for. Genetic testing like the gene machine, it wouldn't even be available if it weren't through this NCBI. It's great that Map My Genes can get you to take a swab of your DNA, of your saliva, send it off, they can isolate that DNA. That's great and dandy, but if they don't have something to compare it to, this NCBI, then they don't have anything. They can't do anything with that genetic information. This is what makes that possible. The other thing I want you to write down is the definition for bioinformatics. Bioinformatics is the application of computer science and information technology to biology and medicine. It is used to create databases, search databases, compare sequences, and represent molecular structure. So there's actually, we're not going to look at this, look into this far, but there's actually a way to see your proteins 3D. And this has helped scientists understand a lot about proteins. We're not going to go that far where we're actually going to design a protein in 3D. We're not going to go that far. But what we're doing here is we're taking all this biological information that scientists have figured out. We know our DNA sequences. We know where we're similar. We know where we're different. Remember the SNPs? Actually, we're going to get into SNPs here in a little bit. The STRs, the single tandem repeats, those kind of things are things that are different, and we know that. And we're taking all of that information that scientists have come, and we're merging that with computer science. We're merging that with the ability to use what we know about computers and technology to huddle that together and then use it in some way. And we're going to look at how we would use this with a case study on breast cancer today. So what are you going to do today? Today you're going to be using a worksheet. You're going to navigate the National Center for Biotechnology Information, the NCBI, and you're going to focus on the BRCA1 gene, so the BRCA1 gene we discussed the other day. You may feel really overwhelmed with the amount of information available on this website, but the purpose of this lesson, remember, is not to learn detailed information about this gene or about its sequence or about this website in general. The big picture here, what I want you to take away from this, is gaining a real appreciation for the depth and breadth of the information that's available through the NCBI. I want you to understand what's out there and how it can be used. Not really that you necessarily are going to memorize any information that you come across today. So Ms. Arnell is now going to distribute your handout. When she distributes it, you're going to take a look at it, and you're going to notice that there are several questions you're going to have to answer. As you're answering them, there may not be enough room for you to answer the questions on this worksheet. Matter of fact, if you notice at the top it says Lesson 2 Class Set, which means that this was designed to be a class set and you write it on your own paper. If you don't have enough room, feel free to write these answers on your own paper. I don't need you to necessarily try to squeeze them in tiny writing all over this. One of the first things that it's going to have you do is just go to the website on your computers, and then it's going to give you a little chance to kind of look at what do you think is interesting on here, or what do you think it might do. And then you're actually going to go up to the search box, and you are going to search for the BRCA1 in the search box. And you're going to find a whole lot of information here about it, and it's going to walk you through exactly what it is it wants you to do with this information and where you're going to go with that. So go ahead. If you have questions, feel free to ask Ms. Darnell as you go. I will leave her with a key to help as well in case you really get stuck as a group. At the end of this lesson today, the other thing that she's going to hand out to you, she's going to hand out your homework. The next thing we're going to be doing is we're going to be using this information in a case study of breast cancer, but I need you to look at the background information overnight. You're going to get the Lawler family pedigree. You're going to read it and complete the pre-work for the lab that we will be doing. So you're going to fill in, read their family, their case study synopsis. You're going to read it. You're going to fill in a pedigree that you think would be sufficient, and then do a couple, Punnett squares that you feel like would fit with this family. And then we're actually going to use their genetic information in this database and in another database to determine whether or not they have the BRCA1 mutation. Have fun.